Get ready to dive deeper into spatial biology with the MERSCOPE Ultra platform. In this video, we will walk you through a general protocol for setting up, running, and maintaining your MERSCOPE Ultra. It is recommended that users perform an instrument maintenance wash before and after every MERFISH experiment to maintain optimal performance of system fluidics. Let's start with the materials you will need to prepare your sample for imaging. Take the sample prep wash buffer and formaldehyde wash buffer from the applicable MERSCOPE sample preparation kit. Thaw the MERSCOPE imaging cartridge in a 37 degrees Celsius water bath with about 2 centimeters of water, or enough to ensure the water level rises to 2.5 centimeters up the outside of the cartridge. Let the cartridge thaw in the water bath for 60 minutes. Ensure the valve does not come into contact with the water. Next, warm up the DAPI and poly-T staining reagent for 10 minutes in the 37 degrees Celsius water bath. Then, gently vortex at the lowest setting and spin down reagents. Keep the imaging buffer activator and RNase inhibitor in a benchtop cooler until use. Spin them down using a benchtop centrifuge before use. Return unused reagents to -20 degrees Celsius storage. Freeze-thaw cycles should be minimized. Aspirate the cleaning solution, clearing premix, or formaldehyde buffer from the Petri dish, ensuring all of it is removed. Wash twice with 5 milliliters or 10 milliliters of sample prep wash buffer, depending on the size of the MERSCOPE slide. Gently vortex the DAPI and poly-T staining reagent tube on the lowest setting to ensure the reagent is well mixed. Add 3 milliliters or 6 milliliters of the staining reagent, depending on the size of the MERSCOPE slide, and incubate for 15 minutes on a rocker. Wash once with 5 milliliters or 10 milliliters of formaldehyde wash buffer. Incubate for 10 minutes. Then wash again with sample prep wash buffer. Proceed immediately to the next step. Click Start MERFISH on the display and allow 1 to 2 minutes for the instrument to initialize. Instrument initialization and experiment configuration can be performed during incubation periods on previous steps to save time. Enter the experimental details, such as name and description. Select the panel-specific MERSCOPE codebook and click Next. Use the Next and Back buttons to navigate through the configuration screens. Specify the imaging depth. If unknown, select 10 microns. For FFPE samples, select 10 microns regardless of tissue section thickness. If the sample was stained with the MERSCOPE Cell Boundary Stain Kit, toggle on Cell Boundary Stains under Additional Stains. If the gene panel contains sequential genes, check the corresponding auxiliary bit and select RNA from the menu. If the sample was stained with MERSCOPE protein stain kits, check the corresponding auxiliary bit and select the illumination intensity. Select either FCXL or FCXS flow chamber. Users have the option to pre-select segmentation parameters. If this option is selected, when imaging is complete, the system will automatically proceed to analysis, bypassing the segmentation preview screen. At the end of the configuration, review the configure summary. If satisfactory, click Next. Open the imaging cartridge lid and remove the old MERSCOPE imaging cartridge from the instrument. Discard it per hazardous waste procedures. Remove the new thawed MERSCOPE imaging cartridge from the water bath and dry the outer surfaces, especially the bottom. Ensure the barcode is free of any liquid and note the barcode number. Slowly invert the cartridge 10 times to mix the reagents. Clean the valve with RNA ZAP solution and 70% ethanol using a Kim wipe. Prepare the imaging activation mix by adding 250 microliters of imaging buffer activator and 100 microliters of RNase inhibitor into the imaging activation mix tube and gently mixing. Pierce the cartridge activation port with a pipette tip. Add the full volume of imaging activation mix and mix the solution by pipetting up and down, being careful to avoid introducing air bubbles. Carefully layer 15 milliliters of mineral oil over the liquid in the cartridge. Insert the activated MERSCOPE imaging cartridge into the instrument, ensuring the valve is toward the back and the barcode is toward the front. Close the imaging cartridge lid and click Scan Barcode. If successful, click Prime Fluidics. Open the flow chamber lid, then unlock, and if present, remove the flow chamber from the previous experiment. Disassemble and clean the components with RNA ZAP solution and 70% ethanol using a Kim wipe. Aspirate the sample prep wash buffer. Place the MERSCOPE slide into the base of the flow chamber. Add the gasket. And assemble the aqueduct and top. Ensure the alignment is correct and twist the top until the alignment mark at the top aligns with the lock position on the base. Spray the bottom of the MERSCOPE slide with 100% ethanol and wipe clean 3 times. Connect the assembled MERSCOPE flow chamber to the fluidic lines on the instrument, starting with the output line, then the input line. Click Wet Flow Chamber to initiate wetting. Orient the flow chamber vertically to allow flow to pull bubbles out of the chamber. If bubbles are visible in the flow chamber or input line after wetting, click Pull More Liquid to remove them. Insert the flow chamber into the instrument, aligning it correctly, and lock it into place. Close the flow chamber lid and click Acquire Mosaic to obtain a low-resolution mosaic. The MERSCOPE instrument will acquire a low-resolution mosaic. You can then select your regions of interest using the touchscreen or mouse. Regions can be renamed by clicking the pencil icon next to each region. These boundaries will be saved and summarized on the screen. The software will provide warnings where regions are overlapped or outside the imageable area. Adjust regions by dragging boundary dots as needed, and use the Visible Intensity range slider to adjust image contrast. Once your selections are complete, click Next to proceed. Clean the immersion oil from the high magnification objective with lens tissue. Clean the underside of the MERSCOPE slide before loading into the instrument. Pipette 75 microliters of fresh immersion oil onto the objective, ensuring there are no air bubbles, and bubbles that occurred have popped. Reinsert the flow chamber into the instrument, aligning it correctly, and lock it into place. Close the flow chamber lid and click Acquire Focus. The instrument will attempt to find the focal plane. When successful, review the experiment summary. If you are happy with your experiment summary, click Start Measurement to initiate the fully automated experiment. The instrument will run the sample and indicate when all measurements are complete. If segmentation parameters were not selected during experiment configuration, after the experiment is completed, segmentation parameters can be selected in the Segmentation Preview window. You may select your desired segmentation and decoding options prior to initiating image processing. When your experiment is complete, the instrument will execute automatic fluidic line washes. Follow the instructions on the screen to empty the waste container and clean the flow chamber. At the end of cleaning, click Done to return to the home page and start your next MERFISH experiment.